I remember a particularly nasty interaction where one of our detractors took me to task for admitting that I did not know by what mechanism ivermectin seemed to be working. And she said...
And if you don't know, it must not work.
Right, exactly. How can you say that something works if you don't know the mechanism of action? And of course, that's not how science works at all. You can absolutely know that something works and not know the mechanism of action, but it appears...
frankly, such a mechanistic approach to science is almost always hand in hand with a reductionist approach and almost always revealing of a relatively small mind who may generate sort of bricks in the wall that already exist in science, but is very unlikely to discover new truths. That is 100% not the way that science has to work. There is good mechanistic science out there, as I think this paper we're going to talk about today reveals. But the idea that if you don't know mechanism, you can't know the thing is functional, no.
Right, and any fool knows this, right? I mean, how many games of ultimate frisbee have taken place entirely on a field with nobody on it who could explain what the relationship is between the Bernoulli effect and the way the damn thing flies, right? No, you just know how to throw the frisbee, right? Mm-hmm. So anyway, knowing how it works is not essential, but it's fantastic when it happens because it allows you to go that much farther. You can extrapolate in ways that you could not necessarily otherwise extrapolate. So anyway, it's great that a mechanism of action seems to have emerged.
Indeed. So we ran across this on Dr. McCullough's Substack. He published a short piece called Ivermectin's Mechanism of Action Against SARS-CoV-2 Described. Shame on the hospital symptoms. Shame on the hospital systems that systematically denied patients and their begging families this FDA-approved Nobel Prize-winning wonder drug. He is referring to this paper that came out in the International Journal of Molecular Sciences, which you can show my screen if you like, Zach. Recently, it's actually end of 2022. And the title is SARS-CoV-2 Spike Protein Induces Hemagglutination, Implications for COVID-19 Morbidities and Therapeutics and for Vaccine Adverse Effects. Okay. I think we had talked about me just sharing a couple of excerpts from the paper before we talk about it.
There was one other thing that was, I don't remember if it's in the paper, it was certainly in McCullough's brief treatment of it, which I wanted to remind people of, because there's been so much effort put into confusing people about ivermectin and whether or not it works for SARS-CoV-2, that I just wanted to highlight this piece of information, which is that ivermectin was known to have broad-scale effects against RNA viruses generally. There's a large number of RNA viruses in which it has been shown to be useful. So to the extent that people are scratching their heads over why a dewormer has an effect on SARS-CoV-2, that's not what it is. This is a highly bioactive molecule that has effects, yes, on parasitic worms, also appears to have effects counteracting RNA viruses, of which SARS-CoV-2 is, of course, one.
Yep.
So
not a lot of them, and we wrote that into the piece that we published on my Substack as well. Yep. And it is a kind of a wonder drug, and there's a reason that it was on the WHO's list of essential medicines, and that's not Roger Daltrey's doing. That's the other WHO, and that's the WHO that has since taken out.
Where did Roger Daltrey come from?
Well, until 2020, when you said the WHO, most people who weren't associated with the World Health Organization or in that sphere somehow would think, Pete Townsend and Roger Daltrey and such. But this isn't a sleight of hand on our part, going like, rock stars think that ivermectin works. No, the WHO, it's still on their list of essential medicines. And I believe it's back on a number of other health agencies' lists of recommended medicines for COVID too. But I did not look into that for
it is actually, I checked before the podcast. I have to delve deeper to see exactly what it means, but it is actually on the NIH site. So
it was on the NIH, so be careful there, because it was on the NIH site for a while, but they basically said, but not really.
Yeah.
So I don't know if something's changed.
Research or something like that. But anyway, it's beside the point. I will point out that in terms of, yes, you can know that something works before you know how it is that it works. that there is also something to be said for the integrative aspect of this, right? You have a bacterium found in Japanese soil. I believe a golf course is the original source.
Yeah, I didn't know that.
But in any case,
that makes it weirder in a way.
It does make it weirder.
Yeah, selective effects from the stuff you have to put on grass to make it into a golf course.
Either that, or it's very tolerant. And Satoshi Omura was at the golf course because it's a place that brings people. I don't know exactly what the explanation would be. Yeah. But the thing that I would point out is there's bound to be a fascinating ecological story underlying the question of why this bacterium has effects against a broad range of creatures. Mm-hmm. In other words, if you studied the bacterium instead of studying the compound, you might find that it has a unique environment in which fending off these things is particularly relevant to some story, I think, that we don't yet know. Mm-hmm. And I would love to hear that story. Yeah. How is it that some creature as inconspicuous as this one came to have all of these effects that we find so useful?
Right. And without understanding why, or even what many of the effects were, the discovery by Umura won him a Nobel Prize back in
2015.
So not that long ago, actually, even though the discovery was many decades before. That is often the case with maybe always the case with the Nobel, right? Yep. At least in physiology and medicine, that the award gets granted often decades after the discovery in question.
Decades
I think
after.
all of the sciences, maybe peace not so much, and maybe literature not so much, although literature tends to be across for a lifetime. Anyway, I digress.
Yeah.
Yeah.
Neither here nor there.
Okay, so this article, published in the International Journal of Molecular Sciences, finds that, and you may show my screen if you like, Zach. This is just the second half of the abstract. The results of these experiments were, first, that spike protein from these 4 lineages of SARS-CoV-2 induced HA, which is hemagglutination assay. I want to say that has to be assay, although the way they've got this, that's weird. Omicron-induced HA at a significantly lower threshold concentration of spike protein than the 3 prior lineages and was much more electropositive on its central spike protein region. IVM, that's ivermectin, blocked HA when added to red blood cells prior to spike protein and reversed hemagglutination when added afterwards. These results validate and extend prior findings on the role of glycan bindings of viral spike protein in COVID-19. They furthermore suggest therapeutic options using competitive glycan binding agents such as ivermectin and may help elucidate rare serious adverse effects associated with COVID-19 mRNA vaccines, which use spike protein as the generated antigen. So that's highly technical, as abstracts will tend to be, but it clears up a little bit with a few more sections. And for those, glycan is just a polysaccharide. So hemaggglutinating property basically just means clumping. Okay, so this clumping of red blood cells. This hemagglutinating property of SARS-CoV-2 has important clinical consequences. First, with trillions of red blood cells each circulating through narrow pulmonary capillaries about once per minute, even small dynamically aggregating and disaggregating red blood cell clumps, as can form even in the absence of pathogens, can impede red blood cell oxygenation. Peripheral ischemia, endothelial damage, and vascular occlusion are indeed frequently observed in serious cases of COVID-19, as reviewed in these references. In COVID-19 patients, damaged endothelium of pulmonary capillaries is often observed adjoining relatively intact alveoli, while hypoxemia is manifested despite normal breathing mechanics. These morbidities of COVID-19 parallel those of severe malaria, in which clumping of parasite-infected red blood cells to other red blood cells via don't remember what that is, terminal residues and endothelial cytoadhesion also often result in fatal outcomes. So they are saying this looks similar to malaria. We now are seeing what SARS-CoV-2 is doing, which is it's causing red blood cells to clump, the word being hemagglutination. And we, the authors of this paper, are putting together that thing that we are seeing with the observation, the clinical observations of how it is that people suffering from bad cases of COVID are manifesting in terms of their symptoms.
So I don't know if you want to continue there or I want to add a couple of things. So to translate some of that, it is counterintuitive that capillary beds are actually basically composed of arterioles that are so tiny that basically the blood cells go through close to single file, and any clumping causes these things not to be able to get through. And what they're saying is that that results in damage from these tissues not being perfused with oxygen, and any tissue that is denied oxygen long enough basically ends up dying, which they're saying matches some of the pathologies that we see from a severe COVID infection. And they also say that the lungs are failing to engage in gas exchange even though the mechanics are working. And the basic point is, in order for gas exchange to work, the blood has to get right to this very cusp of contact with the air that you're breathing in. And to the extent that the capillary beds in your alveoli are not perfused with blood cells because the blood cells can't get in single file to get into those tiny little arterioles, it's not working. So they're not presumably dumping carbon dioxide and picking up oxygen, which then results in the hypoxia that was talked about so much earlier in the COVID pandemic.
Yeah, just a semantic point.
Yep.
So, the heart, as mammals, we have a fully four-chambered heart in which we have blood coming out of the heart and going to systemically, coming back in, and then going to the other side of the heart and going out into the lungs, the lungs and then back. And in both of those circuits, be it out to the body and then back, or out to the lungs and then back, you basically go out on the biggest, most vascularized vessels, the arteries. And then as they get smaller, there's no real category difference, but then they become arterioles. And then at the point that they are at their smallest, where the blood cells may in fact be going single file, we call those capillary beds. We don't really call them arterioles, at least over in comparative biology land, where I've never dissected a human, but dissected a lot of cats. And then, as they come out of the capillary beds, like venules, I think, and then veins, as they get bigger, and veins are under much less pressure, and so the sides of them are much less thick, and they tend to, you end up with these sort of pools in your veins potentially if you aren't otherwise very healthy, or if you're old, in a way that you don't really get pools in your arteries, because the blood coming directly out of your heart before it hits capillaries is under fairly high pressure, but after having gone through the capillary beds, it's coming back more sluggishly and also deoxygenated. And the thing about the alveoli of the lungs is that basically the lungs, the pulmonary circuit out of the heart is a way to get the blood back into oxygenated state, right? So, the pulmonary artery is the least oxygenated vessel in the body, or the least oxygenated big vessel in the body, even though in general, when we think of arteries, it's like, the blood is pumping fast, it's full of oxygen, but only if it's recently come back from the lungs and is now going out into the body.
Right. And so, so I did screw up. I used arterial, or I should have said capillary, but in any case, it's amazing how well this actually fits a lot of the stuff that was discussed early in COVID. The hypoxia is basically, if you imagine that what you did, even if your red blood cells are just a little bit tending to stick together, that's enough to keep them from flowing through those capillary beds the way they're supposed to. And so you can imagine they put that pulse ox thing on your finger, and basically what it's reading is that to the extent that oxygenated blood landed in your tissues, like the ones in your fingers, it burned its oxygen and then wasn't in a position to go back and play another round of the game. And so you would see this. Now, what I don't hear explained in here is why you will remember that the syndrome was called something like happy hypoxics. That you had people who had such low oxygenation that they should have been on the floor, and yet they didn't even register to themselves that they were struggling to oxygenate. So I'm waiting for that explanation. Yeah. But anyway, so far interesting.
Yeah. So a few more bits from this paper. This is just something that we've talked about before, but ivermectin achieved Nobel Prize-honored distinction for success against global parasitic scourges, but is of disputed efficacy in the treatment of COVID-19, as indicated, for example, by the disparity in the conclusions of this editorial cited and its key cited meta-analysis cited. So this is these authors, like all the authors trying to publish, all the scientists trying to publish on COVID right now who are in any way dissidents from the mainstream narrative, have to have to do a little bit of this, like, oh, but the vaccines are great. Oh, but, things don't work when we've been told they don't work, even if they really kind of do. And so there's always a little, there's ways that you can get in the critique, and this is them doing that. The conclusions of the editorial that they cite are we need to stop using drugs that don't work. And then the meta-analysis, and that includes ivermectin, and then the meta-analysis that editorial cites finds that it does work. So, even the medical journals that are supposed to be that seem to be doing the right thing are citing things backwards throughout.
Well,
the
you
pandemic.
I guess what you're saying is you can detect the massive force that wants to tell you what you can and cannot say, even in a paper that's reporting something here that's clearly a vindication of those who have been demonized.
But
here.
This is them going like, yeah, look at what these guys did.
The disparity between
but you have to. I'm not going to do it here, but I did before we came on camera. You have to go and look, like, oh, okay, so this is them revealing something that to the casual reader won't be revealed, but it's in here. Okay, so sorry about the scrolling. Where's the next thing? HA, hemagglutinating. Okay, so that's just the tendency of the red blood cells to stick together. The hemagglutinating inducing activity of SARS-CoV-2 spike protein, which is especially potent for Omicron, raises questions as to potential risks for COVID-19 mRNA vaccines, which use spike protein as the generated antigen, even though serious adverse effects linked to spike protein, such as myocarditis, are rare. Detectable levels of SARS-CoV-2 spike protein and S1 in serum or plasma have been found to persist for as long as 50 days following such vaccinations. The possibility that spike protein migrating into the bloodstream could in rare cases prompt such hemagglutinating-associated adverse events is suggested, for example, by a study of 1,006 subjects experiencing adverse events after receiving a Pfizer-BioNTech or Moderna mRNA vaccination, which found a significant degree of red blood cell aggregation in the blood of 948 of those subjects, 948 of 1,006. These risks may be increased for younger age groups, with 301 adolescents of 13 to 18 years of age who received 2 doses of the BioNTech mRNA COVID-19 vaccine in one study having a 29.2% rate of cardiac adverse events, ranging from tachycardia or palpitation to myopericarditis.
Wow. Yeah. So what they're saying, and I should say there's a piece of conservatism in here that doesn't that needs to be highlighted, which is that, yes, the spike protein has been found up to 50 days, but it's actually been found longer than that, and we really don't know at what rate it dissipates.
I remember, I know you're going somewhere, persists for as long as 50 days following such vaccinations. I did not click through on these links, but I remember us talking in an earlier DarkHorse about some of the research that was finding that it was persisting, and the research was saying, up to 50 days or something like that. And when you go into the paper, it actually says, well, we actually just stopped looking after 50 days. So it's not that they are saying, and then it's gone. They're saying, and then we don't know, which is an important distinction. I don't know that's the case here, but it may well be.
It is also the case that people should remember the spike protein is not supposed to be free-floating after these vaccines anyway, right? It's supposed to be sticking in the membrane of the cells that have been transfected, and those cells are not supposed to be anywhere and everywhere in the body. They're supposed to be limited to the deltoid and the lymphatic system associated with it, where the antigen-presenting cells were supposed to pass it off to the immunological cells, the B's and T cells that learn the formula. And anyway, so the point is we're talking about design failure after design failure. The choice of spike protein itself dangerous. Why? Well, we now know at least one reason why, which is that it causes this
hemagglutinating inducing.
It's right. So choice of the spike protein, the fact that spike protein is showing up independent of the cells that were transfected, all of these things are suggesting a kind of blood disorder that is mimicking malaria, and there's a whole other story that we could get into, but nonetheless, the idea that this is creating this overall malfunction of the red blood cell half of the system is profoundly disturbing, and the mechanism of action, therefore, I don't know if you had a piece you wanted to read or if we should just describe it in plain English.
Of ivermectin?
Yeah.
We'll do this first, then we'll translate into plain English.
Good.
Ivermectin, a macrocyclic lactone indicated to bind strongly to multiple glycan sites on SARS-CoV-2 spike protein, blocked hemagglutination when added to red blood cells prior to spike protein, and reversed hemagglutination when added afterwards, which suggests therapeutic options for COVID-19 treatment using this drug or other competitive glycan binding agents. So that's not, that's in a dish, right? But ivermectin blocks hemagglutination before exposure, and it reverses hemagglutination after exposure to the spike protein in a dish, in a petri dish, suggesting that, given that we now know that one of, if not the primary mechanism of action by which SARS-CoV-2 is doing damage to people is hemagglutination. And we know separately that ivermectin, and it's not the only one that does it, but that ivermectin is both incredibly safe and, effective in blocking or reversing hemagglutination. Hey, it works against COVID.
Yeah, it works against COVID. Also potentially works against vaccine adverse events that are caused by spike protein. Yep. I will say it is, if this paper holds, it is a validation of one of the hypotheses that we explored early on, that the mechanism of action of ivermectin might be the binding of spike protein, that might be why it has general applicability. And there was one other thing
I don't think it's, but that's not what's going on here. It's not binding the spike protein.
Well, the question is, so you'll also recall that with Dr. Malone on the episode that I did with him and Steve Kirsch, we talked about the fact that the spike protein is covered in these sugars.
Yeah.
Right? It's covered in these sugars, presumably as effectively a cloaking mechanism, right? So if it is pulling these sugars out of the blood and therefore being encrusted in something that doesn't trigger the immune system, that allows it to evade the surveillance that happens there. And so anyway, it's interesting how this result seems to line up with some of the speculating that we did about why things functioned the way they did early on in the pandemic before this stuff was known. So it's kind of cool.
It's very cool. And it's not misinformation, it's not disinformation, but it's probably still malinformation.
Because it's true and it makes you distrust your government?
Yeah.
Yeah. I'm not sure what we can do about that, really. I mean,
better government, I think.
Better. Wow.
Yeah.
Yeah. You said a mouthful. Right. Yeah. So if we had trustworthy government, then we could at least be emotionally disturbed at things that made you distrust it. But with a distrustful government, it's hard to know if you're just distrusting it because it sucks.
Right. Yeah, you can't know if you're paranoid if they're actually after you.
Exactly. It's very hard to diagnose paranoia when they're out to get you.
No.
Yeah.
Yes. All right.
So
there.
Yeah. I mean, let's just say this is exciting for more than one reason. A, it's beginning to fill in a piece of the puzzle. B, it's operationalizable.
Yeah.
Right. To the extent that people,
which is cool. Mechanism is cool. We don't need it often, but it's cool when you got it.
But it's cool. It's cool when you got it. And I remember, I don't think Pierre said this on the podcast, but he certainly said it to me and maybe us privately, that as he was discovering that ivermectin was not only a great preventative for COVID, a great treatment for COVID, but it also worked for long COVID and adverse events. He was like, this kind of sucks because I feel like a one-trick pony. I keep pointing people to this stuff, but it works.
Right.
And anyway, this begins to explain why it is that it's just sort of generally useful in this, because spike protein is such a large part of the pathology associated with both the disease and the so-called vaccines.
Well, and I think I read it, but I glossed over it because it was a highly technical paragraph. But they also say the hemaggglutinating property of SARS-CoV-2 is much reduced in Omicron. And so quite separately, and I don't know if he said it publicly, but to us, Dr. Corey, when we got Omicron a year ago, he said Yeah, you could take the ivermectin, but it seems to be not as effective with Omicron. And that is also consistent with this finding that Omicron has a lower hemagglutinating tendency than the other strains.
Isn't it crazy when all of the lines of evidence line up?
Yeah.